Welcome to this lecture on biotechnology and its application. What we're going to cover in this lecture is processes of recombinant DNA technology. So for these processes of recombinant DNA technology, we follow this order, and this order is very important because it can be asked for one question out of this order. So first process is isolation of DNA, which DNA isolation from any of the target living organism, which might be a bacteria, plant, or animal cell. Then we cut the target DNA with the help of restriction endonuclease, which is also known as molecular scissors. Isolation of a desired DNA fragment. After that, we ligate the both vector DNA and the target DNA. It will lead to recombinant DNA, and DNA ligase is used for ligating both the end of vector DNA with target DNA. This recombinant DNA will be transferred in the host cell, which might be a E. coli. We culture them into the media, which contains certain micronutrient and macronutrient. Carbon source such as sucrose, etc. Then after that, it will produce some desired product. We will extract those products with the help of certain techniques. Hope that's clear. So let's see these steps one by one. First, isolation of DNA. As we know, DNA is isolating from the bacteria, plant, or the animal cell, right? So we need to break the cell wall present in both bacteria and plant, and in case of animal, only cell membrane is there, not the cell wall. So in case of bacteria, we use the enzyme lysozyme. What it does is it hydrolyzes the bond between NAC, that is N acetyl glucosamine, and NAM, which stands for N acetyl muramic acid. The bond between the two is beta 1,4 glycosidic bond. Lysozyme break this bond and make cell wall unstable, reduce the stability. Hence, we can easily take out the DNA from the ruptured bacteria. After that, in case of plant cell wall is also present, but this cell wall is made up of cellulose, as you can see over here. So this cellulose will be broken up into oligomers or oligosaccharide with the help of enzyme cellulase. And further, beta-galactosidase will break this oligosaccharide into glucose monomers. This is how we break the cell wall of plant. In case of plants, it is very difficult to take out the genetic material. Hence, we can use the mortar pestle for the grinding purpose. So can we easily take out the DNA from the plants. Then in case of fungus, it is chitinase enzyme is used, as the fungus is made up of chitin. Chitin is a polysaccharide, and chitinase enzyme break the chitin. This is the chitin, and easily take out, hence we easily take out genetic material from the fungus. But after getting the DNA or isolating DNA, there are certain impurities come with the cell rupture. Where impurities in the sense RNA or protein. There is a mixture of DNA with RNA plus protein, right? So what we do? So we do certain enzymes will kill this RNA, which is known as RNase. RNase kill will degrade this enzyme, break down this enzyme, RNA. In case of proteins, is degraded by the enzyme proteases. So now DNA and certain debris is there, right? So what we do is we centrifuge the mixture, and after centrifugation some pellet is there which is known as debris, and supernatant is there which containing our DNA. So we take out this DNA with the help of pipette and transfer it to another tube and precipitate it with the help of chilled ethanol. Why we use ethanol? Because DNA is not dissolving in the ethanol, but it get dissolved in the water. Hence we are not using water but the ethanol. So as we know, precipitation of DNA is done with the help of ethanol, chilled ethanol. Let's see the process in detail step by step. First, what we do is we lyse the cell. Lysis of cells will be done, and after that there are some impurities with it, RNA or protein. We use RNase to degrade RNA and proteases to degrade proteins, right? After that we centrifuge the mixture which is DNA and some debris to separate the both. So it will separate in the form of debris pelleted down or settled down, or supernatant containing DNA. After this, supernatant will transfer into another tube which containing chilled ethanol, where the viscous strand of DNA will be formed and it will be spool out. The technique is known as spooling. After this, next step is cutting of DNA at specific location. So this work is done by our restriction enzyme. Right? It will cut the DNA into several fragments. After that, we load it into the agar gel electrophoresis, and this will give us the bag.